Okay, this is the fourth lecture in BIOL1309 Evolutionary Diversity. Now you might remember in the last lecture I was talking about non-vascular plants. That is, plants which lack a vascular system. In other words, they lack the xylem and the phloem. If you remember, the non-vascular plants represent three separate evolutionary lineages near the base of this evolutionary tree. In the last lecture, I spoke about the hepatophyta, and in today's lecture, I'll talk about the other two groups, the anthocerophyta and the bryophyta. Before I begin talking about these individual groups of plants, however, I just want to remind you of the most important points from the last lecture. And I want to begin by reminding you of the life cycle shown by non-vascular plants. All plants, irrespective of whether they have a vascular system or not, have a life cycle that shows alternation of generations. And that's illustrated by this diagrammatic life cycle here, in which there's this horizontal grey line dividing the life cycle into two halves. Here on the left-hand side, we have meiosis, which is cell division accompanied by the halving of the chromosome number. And here on the right-hand side, we have fertilization, which involves the fusion of the gametes to form the zygote. And of course, that fertilization event necessarily results in a doubling of the chromosome number. So within the life cycle, there is alternating doubling and halving of the chromosome number. Now in this diagram, everything above that horizontal line is described as haploid, that is, having one copy of every chromosome in every cell, or having N chromosomes. And everything below that horizontal grey line is the diploid phase, in which there are two copies of every chromosome in every cell, in other words, 2N chromosomes. Now the term alternation of generations refers to a situation in which there is a multicellular haploid phase in both phases of the life cycle. So here it's shown in red and white. We have two balls of cells. This represents the two multicellular gametophytic phases, which are the haploid individuals. And down below we have a pink ball of cells, which is representing the multicellular diploid individual, or the sporophyte. So just to remind you, the gametophyte is literally the gamete producing plant. So these are individuals that produce the gamete. And the sporophyte is the spore producing plant, the plant which produces the spore. This is part of what we call sporic meiosis. And you might remember from the last lecture that it is the haploid phase which is dominant. And by dominant, I mean that the gametophyte is larger and is longer lived. The sporophyte is much smaller, is much more transient, and as we saw in the last lecture, the sporophyte actually remains attached to the gametophyte, and in many cases is pseudoparasitic, is obtaining its nutrition from the gametophyte. Now if we go back to the evolutionary tree, or the phylogeny, we can see that the non-vascular plants represent three different evolutionary lines. In the last lecture I spoke about the hepatophyta, which in English are known as the liverworts. And you might remember that there were two different growth forms. Most species were of a thallose growth form, shown here on the left. And I used a couple of examples, including Marcantia, in the last lecture. But there are a small number of hepatophyta which are leafy, as shown on the right-hand side. But I'd like to remind you that these plants do not have true leaves. These are leaf-like structures, but not true leaves. So what I want to do in today's lecture is to talk about the other two groups of non-vascular plant, beginning with the anthocerophyta. Now, the Anthocerophyta in English are known as hornworts. And this word wort is an Old English word meaning plant. So these are literally horned plants. And they've got that name because of the appearance of the plant. If you look at this photograph, for example, here we have an example of a genus called Anthoceros. What we're looking at in this photograph is both the gametophytic and the sporophytic generations. The large plate-like structure towards the bottom half of this photograph is the gametophyte, so it's a thallose gametophyte. And then these horn-like extensions on the upper surface are the elongated sporophytes. So that one photograph is showing both generations together. Let's have a closer look at the gametophyte first of all. One of the interesting things about the gametophytes of the Anthocerophyta is that if you cut them open, you'll find another organism living inside. The gametophyte is a little bit like a sandwich. There's an upper and lower epidermal layer, but inside that sandwich, there's a colony of what are known as nostoc cyanobacteria. So the word nostoc is a generic name. Nostoc are a type of cyanobacteria, which are also known as blue-green algae, although they're not technically algae at all. Now, cyanobacteria are a very interesting organisms. Here's a photograph showing what they look like, long chains of cells. They're interesting because they have an ability that plants lack. Cyanobacteria are able to fix atmospheric nitrogen. Now, all plants need nitrogen. It's an essential nutrient that they need. But plants are not capable of fixing free atmospheric nitrogen. Plants have to obtain their nitrogen in a derived form, either as a nitrate or ammonia and such like. Now, cyanobacteria are able to fix atmospheric nitrogen. So essentially, these Anthoceros plants are cultivating or farming the nostoc cyanobacteria inside the gametophyte. This is an example of what we call symbiosis. Symbiosis is a mutual relationship in which both partners benefit. The Anthoceros is growing or cultivating the nostoc cyanobacteria. The nostoc is therefore providing the Anthoceros with fixed nitrogen. And the nostoc colonies benefit from growing in a very protected environment. They're encased within the gametophyte and are therefore protected from drying out. Now, if we have a look at the upper surface of the gametophyte of Anthoceros, this scanning electron micrograph down here shows a number of small openings into cavities on the upper surface of the gametophyte. And these are openings into the cavities where the antheridia and the archegonia are formed. If you look at this diagram in the top right-hand side of this slide, here we can see antheridia in a small chamber. And here on the left, we have the archegonia. Now, the antheridia have a very simple structure. They're just bags full of antherozoids or sperm cells, as I described in the last lecture. And the archegonia again have this characteristic flask-shaped structure. The line of cells that I'm indicating here with the cursor reflect the archegonial wall. So we have the egg sitting at the base of the archegonium with a long canal running to the outside. So the antherozoids, or the sperm, are released from the antheridia. They will then swim through rainwater to the top of the archegonium. They will swim down the canal of the archegonium and fertilize the egg to form the zygote. The zygote will then undergo mitotic cell division and start growing into the young sporophyte. And as you can see in the photograph on the left, the sporophyte is this elongated structure which grows out from where the archegonium was. Now, my The next slide shows a very young sporophyte growing. This ball of cells that I'm indicating here is the apex of the young sporophyte. So it's still very short in this photograph on the top right because it hasn't grown very much. All the other cells surrounding that sporophyte are cells of the gametophyte. And this ring of cells that I'm indicating here is the sheath at the base of the sporophyte. So as the sporophyte grows, it will elongate, and these diagrams here show a young and a mature sporophyte. At maturity, the sporophyte has three basic component parts, the same as we saw in the last lecture in Marcantia. Here we have the foot, which remains embedded in the gametophyte. Here we have a short stalk, and the rest of the sporophyte is this very elongated sporangium. And I'd like to point out in this diagram that the contents of that sporangium seem to change at different heights. Here at the top there are mature spores. Each of these small circles represents a spore waiting to be released. But as we go down the sporangium, those cells get smaller and in fact they are less mature. So there's a gradation in terms of the maturity of the cells within the sporangium. The most mature cells are at the apex, and the youngest, most immature cells are towards the base. And we can see the same thing if we have a look at some photographs. First of all, let's have a look at the central portion of the sporangium. This photograph on the right-hand side shows a longitudinal section through the sporangium. So here, down the center, we have all the mature spores. And there's a sporangial wall on the left here and on the right. Now, one feature I'd like to just point out to you is this structure here, which is actually part of a gaseous exchange pore. Now, we heard about gaseous exchange pores in the last lecture, and of course they're closely tied with photosynthesis. So for photosynthesis to take place, the plant needs to capture carbon dioxide. And that carbon dioxide is removed from the atmosphere through these gaseous exchange pores. So here we have a sporophyte which has a gaseous exchange pore, so it's clearly something which is undergoing photosynthesis. And in that context, I'd like to just point out that this sporophyte here on the left is green, is photosynthetic. So in the last lecture, I talked about sporophytes as being pseudoparasitic, deriving their nutrition, their food, from the gametophyte. But here in the Anthocerophyta, to some extent at least, the sporophyte is able to generate its own food. It does still remain attached to the gametophyte, but it's at least able to create part of its own food. So in this upper portion, or the middle portion of the sporangium, we have mature spores. And if we look at the base of the sporangium, well, we can see something very different. Here is the foot, which is remaining embedded inside all the cells here of the gametophyte. This region here is the short stalk. And these cells here at the very top of this photograph are the cells at the base of the sporangium. And as I mentioned earlier, there is a gradation in terms of the development of the cells inside the sporangium. So those two photographs can be linked together with the dotted lines here representing regions that have been removed. So we have very mature cells further up and very immature cells further down. So different levels within the sporangium reflect different developmental stages. And we can show that graphically here. Down at the bottom of the sporangium, we have cells which are described as myocytes. Now, a myocyte is also known as the spore mother cell. And I think that explains what those cells do. They're cells which undergo cell division to create the spores. And if we look a little bit higher up in the sporangium, we have cells which are older, are more mature. And instead of myocytes, we have spore tetrads. Now, the word tetrad refers to a cluster of 4 spores that came from the same maternal cell. So if you like, these are 4 immature spores. And further up in the sporangium, we have the mature spores. They separate from their tetrad and become 4 independent spores. Of course, within this sporangium, there are many hundreds of sporocytes, so there are even more spore tetrads and more spores. But there is this gradation in terms of the maturity of the cells inside the sporangium, with the mature spores at the apex and the immature cells down at the base. So how can we explain this gradation of maturity of cells inside the sporangium? Well, I think there's an ecological explanation, which becomes apparent if you look at the way in which the sporangium opens up. Now, unlike the sporangia we saw in the last lecture, which does kind of burst open at maturity, in Anthoceras, the sporangium splits open from the apex. It's a little bit like a banana splitting open from one end. Splits open and the split extends from the apex down towards the base. It splits as the cells become mature. So when only the very apical spores are mature, then the sporangium is only splitting open at the very, very top. And it's split towards the base as the wave of maturation of the cells progresses from the apex to the base. So from an evolutionary point of view, this is a very good strategy because it means that the spore release will be extended over quite a prolonged period. And that means that at least some of the spores will be released at the ideal time. And the ideal time is when the air is very dry because these spores are being dispersed by wind. If the air is damp, it means it's been raining, and rain, of course, would wash the spores out of the air. I'd now like to move on to the last of the 3 groups of non-vascular plant, which are the Bryophyta, in English known as the mosses. And this is perhaps the group of non-vascular plant you'd be most familiar with. If you go hiking in Hong Kong, you will doubtless have seen these plants growing either on the ground or on tree trunks or on rocks near waterfalls and so on. They are small plants, normally growing only a few centimeters high. They normally have erect growing stems with small leaf-like structures. And the photograph on the left here has these pin-like structures growing from the upper surface. And as I guess you've probably already realized, those pin-like structures represent the sporophyte. So the green structures with the leaf-like organs down below are the gametophytes, and these pin-like structures are the sporophytes. Now, if we look at the classification of the Bryophyta, we can group the members into 3 different classes. Now, a class is a taxonomic unit. Now, most of the Bryophyta belong to the class Bryidae, in English simply known as the true mosses. And I'm going to spend most of my time talking about the Bryophyta, actually discussing the Bryidae. But the other 2 classes are the Sphagnidae, which in English are known as the peat mosses, and the Andreidae, which are known in English as the granite mosses. Now, before I talk about the appearance of these 3 different classes, let me just explain what it is that unifies all these 3 classes as a single group, the Bryophyta. What is the characteristic feature of the Bryophyta? Well, it's the possession of a thing called a protonema, and a protonema can be defined as an immature gametophyte. Now, I think the best way of explaining the protonema is to have a look at some photographs. And we can start with this photograph here, which shows a spore of the bryophyta germinating. So this brown structure on the left-hand side here is the spore. And this thing coming out on the right-hand side is the thing that's germinating from that spore. So basically it's a cell growing out. And these green blobs inside that cell are the chloroplasts, which of course contain the chlorophyll. Now if we watch what happens to that structure, it will grow and develop into this long thread-like organism. Now, I don't know if you've ever looked at fungal hyphae under a microscope, but fungi spend a large part of their life cycle growing as thin thread-like structures. It looks very, very similar to this structure here, which is what we call a protonema. Now we know that this is not a fungus, of course, because of all these green blobs, these chloroplasts. Fungi are not plants, they do not have chloroplasts, they do not have chlorophyll. So this is clearly a plant, but it's growing in a form that looks a little bit like a fungus. This structure, which is known as a protonema, will grow and extend over the ground, and then at some stage it will germinate and produce the mature gametophyte. And the mature gametophyte has a little stem with these leaf-like structures, as we've seen before. So the bryophyta is quite distinct from the other groups of non-vascular plant because it has this immature gametophytic phase known as the protonema, a phase which looks a bit like a fungal hypha, but which is clearly not a fungus because it has these chloroplasts. Okay, so let's have a look at the three different classes of bryophyta. And I'll begin talking about the Bryeidae, and I'm going to spend most of my time talking about the Bryeidae, and I'll only come back to talk about the Sphagnidae and Andreidae very briefly towards the end of this lecture. So let's have a look at some photographs of typical examples of the Bryeidae. Here is a plant probably grows no more than 3 or 4 centimeters high, but it has these little erect growing branches with spirally arranged leaf-like organs. Some members of the Bryeidae don't grow erect, but rather grow horizontal. So this is an example here. It looks a bit feathery in appearance, but it still has stems with leaf-like organs arranged. What we're looking at here, of course, are the gametophytes, or the multicellular haploid phase of the life cycle. So these are plants which are bearing the gametes in Antheridia and Archegonia. And those Antheridia and Archegonia are actually located towards the apex of the stem. So in this photograph, we can see these leaf-like organs, but towards the apex of the stem, those leaf-like organs look a little bit different because in amongst those organs, there are the Antheridia and Archegonia. Now my next couple of slides show a longitudinal section through the apex of one of these stems. So if we were to cut vertically and take a very thin slice through that plant, this is what we might see. And in these photographs I'm going to show you here, I want to emphasize that the plant tissues have been stained using stains which highlight the cell walls, so they're not the natural color. Now all the tissues we're looking at here are part of the gametophyte. The cells at the base here are the top of the stem, and we have these leaf-like organs coming off. There's one there and one there. But what's very obvious in this photograph are these very dark staining structures, and I hope that you've guessed that these are the Antheridia. These are very similar to the Antheridia that we saw in the last lecture. Antheridia are basically just bags full of sperm or antherozoids. So all this speckled tissue here are the sperm waiting to be released. They're protected by this antheridial wall, stopping it from drying out, and there's a very short stalk at the base. Now, the only other structure in this photograph which is noteworthy are these structures in the background here, which I have labeled here paraphyses or paraphyses. I think you can choose your pronunciation. Now, I'll come back to explain what the function of these paraphyses might be in just a moment. But what we're looking at in this photograph is a section through a male gametophyte. It's a gametophyte which is only producing sperm or antherozoids in antheridia. This next photograph shows an equivalent longitudinal section through a gametophyte which is bearing archegonia. And again, I'd just like to emphasize that the colors here are not natural. These are stained plant preparations. But there's a whole series of organs there that I hope you can recognize as an archegonia. The archegonia have the same basic structure that we saw in the last lecture. Here, for example, we have the egg waiting to be fertilized. It's protected by the archegonial wall. Above the egg, there's a long neck which has a canal running down it. And of course, the sperm will be released from the antheridia. They'll swim through rainwater towards the apex of the archegonium. The sperm will then swim down the neck and fertilize the egg. Now, the only unusual structure about this archegonium is its rather elongated stalk at the base. But apart from that, these archegonia look very, very similar to the ones we saw in the previous lecture, for example, in Marcantia. Now again, we have these structures in the background here, which I am labeling the paraphyses or paraphyses. And I mentioned a moment ago that I would try to explain the function of these organs. In fact, botanists are not very sure what those organs do. There's a couple of different hypotheses that have been put forward. The first hypothesis is that they are there for protective purposes. They may protect the archegonia and protect the antheridia. And by that I mean protecting against desiccation and drying out during their development. Another hypothesis, however, is that these paraphyses don't actually have any function at all. Some people have speculated that these paraphyses might represent antheridia or archegonia that failed to develop into reproductive organs. So all the antheridia, archegonia, and paraphyses initiate as small organs, and whether they develop into an antheridium or an archegonium or remain as a paraphyses are largely dependent on the receipt of plant hormones during development. After the egg has been fertilized, it forms, of course, a zygote, and the zygote then begins to undergo mitotic cell division to form the embryonic sporophyte. And the sporophyte then grows up and out from the gametophyte to form these pin-like structures. So each of these structures is a sporangium on a stalk, and at the base is a foot connecting to the gametophyte. Now in this photograph, I'm looking at 5 different developmental stages in the formation of the sporophyte. So at the bottom here, this green structure with the leaf-like organs, of course, represents the gametophyte. And here we have the sporophyte growing from where the archegonium was at the apex of that stem. Now the youngest specimen here on the left is already very highly developed. There will already be a sporangium, a stalk, and a foot, the 3 main parts of the sporophyte. But the final stage in development is the elongation of the stalk, and that's what we can see happening in these 5 different specimens. suddenly the stalk is elongating. Now what you're looking at in that brown structure on the left-hand side is not actually the sporophyte itself. That brown structure is actually the archegonium. Because if you remember, the archegonium grows and expands to surround the young developing sporophyte. So the sporophyte is protected by the archegonium. So that enlarged archegonium contains the highly differentiated sporophyte. But then the elongation of the stalk has the effect, the consequence of tearing the top of the archegonium off. So this brown structure here is actually the upper half of the archegonium. It's been torn off and borne upwards. And that part of the archegonium is called the calyptra. It's a papery hat which is derived from the enlarged dead archegonium. And I think perhaps that is clearer in this photograph. Here we have a whole cluster of sporophytes from the Bryonidae. This brown papery structure here is the top of the archegonium, it's the calyptra. At the base here we can see that clearly has been torn away. There are a jagged base where it's been torn from the rest of the archegonium. Now these calyptra can be removed possibly by passing animals brushing past the plant or by wind blowing them away. But when the calyptra is removed, it doesn't result in the release of the spore, because the sporangium, which contains the spores, has its own hat here, a structure there on top of the pot of spores, preventing the spores from being released. And we can see that here. This photograph on the right-hand side shows a longitudinal section through the sporophyte of a member of the Bryonidae. Now this particular specimen has a very, very short stalk. So this structure at the bottom here is the foot where it remains embedded in the gametophyte. This structure here is the stalk, which remains very short in this specimen. This is the sporangium here. And all these dark brown staining cells here are the spores waiting to be released. And at the top here we have the hat, this little lid. The technical term for that is the operculum. So the spores cannot be released until that operculum lid is removed. And the operculum lid will come off when the air is very, very dry. But even after the operculum has been removed, the spores are still not free to leave the sporangium. Because after the operculum lid comes off, the opening at the top of the sporangium is blocked by a series of inwardly pointing teeth. So all these teeth here, which look a little bit like the legs of an octopus or a squid, are arms which can fold backwards. So as the air dries, these arms fold backwards, allowing the release of the spores. And that's actually a very good mechanism for controlling spore release, because the spores are only being released when these teeth are folded outwards, and they only fold outwards when the air is dry. And if it begins to rain and the air becomes quite moist, these teeth will fold back again and to stop the remainder of the spores from being released. So these teeth are a way of regulating the release of the spores from the sporangium. And here's a photograph showing these teeth, and if you like terminology, the correct term for these teeth are the peristome teeth. Now, when I'm talking about different groups of plant, I always like to summarize what we've learned by looking at the life cycle. So here is the life cycle of a typical moss. So here I'm not giving a specific name of a genus. This is just a generalized life cycle for a typical moss. But as always, the life cycle is divided by this horizontal line. Here we have meiosis on the right-hand side, which is cell division accompanied by halving of the chromosome number. And here on the left we have fertilization, or the fusion of two cells resulting in doubling of the chromosome number. So everything below this horizontal line is the haploid phase of the life cycle with N chromosomes in each cell. And everything above that horizontal line is the diploid phase of the life cycle with twice as many chromosomes, 2N chromosomes in each cell. So let's have a look at the life cycle, beginning with the multicellular haploid phase, or the gametophyte. And here we have the female gametophyte and the male gametophyte, both elongated structures with these leaf-like organs, and towards the apex we have the antheridia and archegonia localized. Here's the top of that stem, and here is an antheridium. And here are the archegonia having their normal shape with a long neck with a canal running down it, and the egg embedded deeply within the archegonium. Now the sperm are released from the antheridia. The sperm are flagellate, they have flagellae which enable the sperm to swim, and they swim through rainwater to the apex of the archegonium and then swim down the archegonium to fertilize the egg. That forms the zygote. And the zygote is shown here in green, and it's put above this horizontal line because, of course, that is now a diploid cell. And that zygote will undergo mitotic cell division and form the sporophyte, which has this three-part structure: a sporangium on a long stalk with a foot at the base connected with the gametophyte. And in this diagram we have the calyptra, the papery hat, sitting on top of the sporangium. There it is shown in the larger scale. It has a torn lower margin where it was broken away from the base of the archegonium. And underneath that calyptra we have the operculum, a lid that blocks the top of the sporangium. And here we have the peristome teeth regulating the dispersal of the spores. And inside the sporangium we have the spores themselves. And the spores here are drawn below that horizontal line because, of course, they are created by meiosis. And these spores here are germinating to form this immature gametophyte known as the protonema, which looks a bit like a fungal hypha, but with these chloroplasts floating around. And after the protonema has grown for some time, it will then produce a bud, which germinates to form the mature gametophyte. Very briefly, to close this lecture, I want to talk about the Sphagnidae and the Andreidae. And I don't want to say too much about these two different classes because they're not as important as the Bryeidae. But I would like to just show some photographs so you know what the plants look like. And I'd also like to just highlight a couple of interesting features about each of them. And we'll start off with the class Sphagnidae. And I'm going to use the example of a genus called Sphagnum. Now, Sphagnum doesn't look very interesting. It's a small, erect growing plant with leaf-like structures, actually very similar to the Bryeidae. But what I think is interesting is when you look at the structure of the sporophyte. So in this photograph, of course, we've got the sporophyte and we've got the gametophyte. The gametophyte has these leaf-like structures. The sporophyte is actually looking very similar to a little Chinese teapot with a little operculum lid there. Now, so far, things don't look very different from what we just seen in the Bryeidae. But these structures, these green structures here, which look like they might be the stalk of the sporophyte, are actually not part of the sporophyte at all. They're part of the gametophyte. The brown structure, which looks like the teapot, is the entire sporophyte. So the foot of that sporophyte is somewhere about there, and this elongated green structure is part of the gametophyte, raising the small sporophyte up high. So the term given to that structure is a pseudopodium, which literally translates as false foot. It's a way of raising up the sporophyte so that when the spores are released from the sporangium, they're held up high and can disperse more easily. And since I spoke about the spore dispersal mechanism in the Bryonidae, let me do the equivalent for the Sphagnidae. So here we have a diagrammatic representation of the sporophyte. It's a sporangium with an operculum sitting on top of the gametophytic pseudopodium. But as the air around the sporangium dries out, the sporangium will start to dehydrate and will want to shrink. But because of the shape of the sporangium, it can't shrink very easily. Pressures will build up until the pressures are so intense that the sporangium will suddenly collapse. And that's what you can see happening in this diagram in the middle. And this sudden collapse of the sporangium will have the effect of blasting the operculum lid off so that the spores are explosively discharged. So here we have a very different mechanism of spore dispersal. Here, spore dispersal is being achieved at the time when the conditions for spore dispersal are ideal. And the last group of bryophyta I want to talk about are the class Andreidae, which in English are known as the granite mosses. And again, I don't think the plants are terribly exciting to look at. Here's an example, the genus Andrea. In this photograph, we're looking at both the gametophytic and the sporophytic generation. So this is the gametophyte, and here we have the sporophyte. And I'd like you to have a look at the structure of the sporangium here. I think things are a little bit clearer in this diagram here. So here we have the gametophyte with its leaf-like organs, and here we have the capsule or the sporangium on a stalk with the foot somewhere down there. And this little inverted Y-shaped structure represents the calyptra. It's the top of the archegonium. Now if you have a look at this diagram of the sporangium, we can see several vertical lines, and these are lines of weakness. And you can see that those lines don't extend to the top or the base of the sporangium. So when the air gets dry and the sporangium has reached maturity, it will start to shrink. And it will shrink by collapsing top down. And it does this by splitting open at the lines of weakness. And this happens explosively, resulting in the release of the spores. And my final slide in this lecture is a scanning electron micrograph showing what the sporangium looks like after it's collapsed. So there are a series of lateral openings, which is where the sporangial wall has split, explosively discharging the spores. But the sporangial wall is connected at the base and at the apex, so it's collapsing top down. And that's the last slide in this lecture, so I'll end at this point.